Hi everyone, I am Dave Beatriz Valera and welcome to mitosis and meiosis cell division simulation video lesson. But before we jump to the actual process or the step-by-step process of the M phase or the division phase, let us first discuss the prior stage and the process before the cell undergoes division. The cell cycle is composed of two main stages, the interphase and the M phase or mitotic or meiotic phase. The interphase is known as the preparatory phase where the growth and the development of the cell happens, while in M phase or mitotic or meiotic phase, it is the division process of the cell. Interphase is divided into three stages, the G1 phase, S phase, and G2. And then, it is now ready to go to the M phase. The first phase within interphase is called the G1 phase, also known as the gap phase between the cell division and the DNA replication. Now, this G1 phase is also known as the longest phase or stage of development or where the growth happens, such as these biosynthetic activities of the cell happens during this period or phase, which is the G1 phase. Now, the G1 phase ends when the cell reach its maturity. Now, the next phase would be the S phase, which stands for synthesis. Now, after the DNA synthesis or synthesize and the genetic materials have already been reached or have already been undergone the growth and development period, the DNA now will start to duplicate or to replicate. Now remember that in S phase, the DNA will start to duplicate or replicate. Now the last phase for interphase would be the G2 phase, which also lasts at the end of S phase until the cell enters mitosis. Now remember that during this phase, there is a significant activity or happenings on the biosynthesis of this cell, like in production of microtubules, and a cell that is ready to enter the mitosis is called a mother cell. Now after the cell undergoes the interphase, which is the preparatory phase, the cell now is ready to enter the M phase or the M phase, which may take place by mitosis or meiosis. Now mitosis and meiosis have also these phases which we will be tackled later on the simulation proper. Now, to remember these phases, let us use this mnemonic device, which is PMAT. P stands for prophase, M stands for metaphase, and A stands for anaphase, T is for the telophase, and the additional one is C, which is the cytokinesis or the division proper of the cell. Now I will show you the stages or phases and the process of mitosis. And for this activity, we will need these following materials. This would be the nuclear envelope or nuclear membrane of the cell. This would be the nucleolus of the cell. And these two pairs will be the centrioles for the opposite sides of the cell. And these two would be the chromosomes, the paternal and the maternal, with this white tape as the centromere holding the two sister chromatids together. And now, we're ready to go. Now imagine that this bond paper is the cell membrane. In prophase, chromatids are starting to condense and become visible as these chromosomes with or composed of two sister chromatids that are held together or bind together by the centromere or at the centromere. And note that centromere has a special proteins that attach, that there is a special protein attached at the centromere to form kinetochore. after that, this nuclear envelope will now disintegrate, and this nucleolus will disappear. And now, the centrioles or centrosomes are free to move. That's why they are now moving from the opposite sides or opposite poles of the cell. The spindle fibers will now start to form. And this would be the prophase of mitosis. In metaphase, the spindle fibers will now align these two chromosomes at the center of the cell, which is called the metaphase plate. And during this phase, the sister chromatids that are held together are repelling each other, forming this familiar X-shaped chromosomes. This formation or organization of these sister chromatids helps to ensure that on the next phase, which is the anaphase, when the chromosomes are separated, each chromosomes will receive, or each nucleus will receive one copy of this, or one copy of each chromosomes. In anaphase, enzymes break down the proteins at this centromere, allowing these two sister chromatids to separate from each other. And once they are separated, they are now considered as a single chromosomes or a haploid chromosomes. These two will now separate from each other and moving at the opposite direction or opposite poles of the cell. Now in telophase, after these two chromosomes reach the opposite poles of the cell, a new nucleus will reform around the chromosomes, forming two daughter nuclei. Inside this nuclei, these chromosomes will start to uncoil. They will start to uncoil, and the nucleoli will start to reappear. The nucleoli will start to reappear in this cell, and outside of this cell, the spindle fiber will disappear. And these chromatids or the chromosomes, uncoiled chromosomes, will now become invisible at the naked eye of humans, or invisible rather in the microscope. And this would be the look for the telophase. It is now ready to perform the actual division, which is the cytokinesis. Once the nuclear contents have been duplicated, the cytoplasmic contents, organelles, membranes, proteins, and the cytoskeleton, the cell now must be divided. And that ends the process of cytokinesis. That ends the process of mitosis, forming these two daughter cells with these equal chromosomes, and the cell now is ready to perform another cycle. Meiosis 1 prophase 1 is just like in mitosis prophase, where this DNA or the DNA is condensation. The DNA condensation is occurring or happening, and this nuclear envelope will disintegrate. This nucleolus will disappear, and these centrioles or centrosome will move at the opposite sides of the poles. This spindle fiber or the spindle fiber will now form like that. But the big difference is what is happening on the chromosomes itself. Like in prophase 1, chromosomes are visible as tetrads, or they are forming like this one. They are forming like tetrads, or they are forming tetrads, and in this process, this is now ready to perform the crossing over or the crossover of their genetic information or genetic materials, the DNA recombination of chromosome. In prophase 1 and metaphase 1, the genetic recombination is occurring. That is right. In metaphase 1, the genetic recombination or the genetic recombination process is still ongoing. And when it enters now in anaphase 1, the genetic recombination is over. Remember that. Now, in metaphase 1, with the help of the spindle fibers, this will now align these tetrads into the metaphase plate like this. And it is now ready to anaphase 1. In anaphase 1, tetrads will pull apart and the chromosomes with two chromatids will move or go to the opposite sides or opposite poles of the cell. In telophase 1, two chromosomes or chromosomes with two chromatids will decondense and the new nuclear envelope will reform around them and the nuclei will reappear. And it will look like this. The spindle fiber now will disappear. Each nucleus now is haploid. The centrosome or centrioles are duplicated and that ends the process of telophase. This is now ready to enter the next stage of meiosis 2. Now in meiosis 2, prophase 2, chromosomes with two chromatids become visible. The nuclear envelope and the nucleoli will disappear. And these two centrioles or centrosomes will move from the opposite sides or poles of the cell, and the spindle fiber will start to form. In metaphase 2, the chromosomes with two chromatids line up at the equator of the cell or the metaphase plate of the cell. With the help of these fully formed spindle fibers. Now remember that in meiosis 1, the genetic recombination is happening at prophase and metaphase 1, but in this metaphase 2, this formation of chromosomes also help in the process of making a unique daughter cells. In anaphase 2, chromosomes will split so that chromosomes with only one chromatid will move or will go to the opposite poles of the cell. They will now split and go to the opposite poles of the cell. Now in telophase 2, these spindle fibers will now disappear and chromosomes with only one chromatid condense and get surrounded by a new nuclear envelopes and a nucleoli. And now it is ready to perform the next phase, which is the cytokinesis or the actual division of cells. And this will form or divide by 4 daughter cells with and all have a haploid chromosomes or chromatids that has the right amount of DNA, and they are now ready to develop a sperm into a sperm or an egg, and it will look like this. Each cells have this haploid chromosomes. a nucleoli on each cell, and these centrioles. And these cells now are ready to perform another cell cycle. That ends the process of meiosis 1 and 2.